motors are devices that can use one form of energy and convert it into mechanical energy. Motor at your house, for example, runs on electricity. The engine of your car is also a motor that runs on petrol. No matter what the initial source of energy is, ultimately the output is in form of mechanical energy. In terms of molecular motors, we have already talked about two molecules that do this job without without classifying them as motors. For example, DNA polymerase walks along the track. It is converting one form of energy into mechanical energy because it is moving. The motion is due to mechanical energy. RNA polymerase, for example. But here we are going to talk about motor proteins that move along cytoskeletal elements, microtubules or actin filaments. So let's look at motor proteins. Molecular motors harness the chemical free energy released by hydrolysis of ATP in order to perform mechanical work. Let me point out, in terms of energetic efficiency, molecular motors are superior than any currently available man motors. Motor proteins associate with their filament tracks may it be microtubule or actin. As we know, intermediate filaments are not non-polar structures, so they do not associate generally with motor proteins. The actin filaments or microtubules. These motor proteins interact with these filaments through their motor domain that binds and hydrolyzes ATP. Here on the diagram, let me point this out. If you look at this structure, this would be the motor domain of this protein. So this protein has two motor domains, and also it has a domain that can bind the cargo, whatever that cargo may be. It may be a mitochondria or it might be a vesicle. We have talked about vesicle transports. So these motor proteins can bind microtubules or actin filaments on one end, their motor domain, and their cargo domain binds whatever the cargo is. The coordinated nucleotide hydrolysis and conformational change cycle causes motor proteins to switch states between strongly bound to their filament tracks or be unbound. In this diagram, for example, you have this motor protein. The two heads, although they are identical, just to identify which head is in the forward position or attached to the filament and which head is not, we have colored them differently. Here we have a light gray and also a black head. This motor protein has two motor domains. In initial form, motor protein is in relaxed configuration. The light gray head is attached, and it is behind the black gray, and it is behind the black head. So there's a conformational change. May it be ATP hydrolysis that caused it or something else. It causes both these heads to bind, and also it introduces a strain in this molecule in the form of overwound or twists this molecule. When this energy is released, the gray head, which was previously behind the black head, now it comes in the forward position. Again, there's a conformational change, and now the gray head is bound, whereas is bound to the filament, and also the black head is bound to the filament. But then this whole will reverse and get back to this state. So this process repeats over and over, which results in movement of motor proteins taking little steps, carrying their little cargo along with them. I would like to point out that motion is a relative term, and as motors can move cargo along these actin filaments or microtubules, if we fix motor proteins on a solid surface and place these filaments, actin filaments or microtubules, whatever that protein interacts with, and provide ATP energy, now rather than motor proteins moving because they're fixed, they cannot, the filaments, actin filaments or microtubules will move along the surface of the structure where these motor proteins were attached. Now I will talk about motor protein that interacts with actin filaments. Myosin, you have all heard about it. It's a skeletal muscle myosin called the Myosin II. This protein, it's an elongated protein, has two heavy chains and two copies each of two light chains. So here you can see this green structure, light or dark green, this is basically the heavy chain, and these two small blue protein structures, they are basically the light chains, and we have two different types of light chains, one shaded light blue and other the dark blue. The heavy chain is globular. It's a head domain. It is present at the N terminus of molecule. This is the force generating machinery, which is followed by a long amino acid sequence that forms extended coil region that mediates heavy chain dimerization. So these heavy chains, they form dimers, which is mediated by coil alpha helices. The two light chains bind close to the head domain, and the long coil tail bundles itself with the other myosin molecules. So as we'll see later, these myosin molecules, they aggregate together and form filaments themselves. Now, let me show you that structure. Tail-tail interactions result in formation of bipolar thick filaments. What do we mean by bipolar? Here, this part of this filament has myosin heads oriented in such a way that they will, if this is an actin filament, this is the minus end, and if this is the plus end, this filament will move in this direction. The polarity is sort of switched on this side because these heads are arranged in such a way that if they will move, they will also move towards the actin filament, but they'll move in this direction. So they're bipolar, and since they're thick, they are also referred to as thick filaments. When we talk about muscles, we'll refer to them. They have hundreds of myosin heads oriented in opposite direction at the two ends in these filaments. Each myosin head binds and hydrolyzes ATP. Using the energy of ATP hydrolysis, it can walk towards the plus end of the actin filament. The opposing orientation of heads in thick filament makes filament efficient in sliding pair of actin filaments which are oppositely oriented past each other. What we are saying is, so you have actin filaments here. If you have, imagine you have an actin filament here and you have an actin filament here. This is the plus end of this actin filament, and this is the plus end of this actin filament. So when these motor proteins are activated, myosins are activated, they'll move this filament in this direction and this filament in this direction. So this is how the thick filament or myosin filament is organized inside cells. Myosins are a very diverse group of proteins. Most myosins are found in all eukaryotes, and this fact suggests that myosins probably arose early in eukaryotic evolution. There are some exceptions. For example, myosin 8 and 11 are only found in plants, but others are fairly common. Yeasts, for example, have 5 myosins, 2 type 1, 1 type 2, and 2 type 5 myosins. This information suggests that these 3 myosins are probably necessary for eukaryotic cell to survive, and the other myosins are have more specialized functions in multicellular organisms. The nematode, which is type of worm we have talked about, C. elegans, for example, has at least 15 myosin genes representing at least 7 structural classes. The human genome includes about 40 myosin genes. Myosin II is always associated with contractile activity in muscle and non-muscle cells. All these myosins, except one, move towards the plus end of actin filament. Although they do so at different speeds, there is only one exception to the rule that myosins move towards the plus end of actin filaments, and the exception to the rule is myosin 6, which moves towards the minus end of actin filament. So this is the diversity. Now let me show you a little experiment. So you have heads of myosin attached to stable structure, a solid structure. It could be a glass slide, for example. And to this container, if you add actin filaments and ATP, this results in movement of actin filaments gliding along the heads of myosin. These actin filaments, how do we know they are moving? you can label them through different procedures. And these actin filaments, if you follow the panel, you will see these filaments are moving. Actin filaments are moving. They're moving along the heads of myosin heads, which have been attached to a solid, stable structure.